All right, so today I will be walking you through an order that I recently put in. I had 13 samples that I wanted sequenced, and when you open the order results page, this is what it looks like. And so you can see right away whether something was unsuccessful. So for instance, this sequence, this individual that I had here, right, for instance, this is the ID number that you put into the order form paired with the primer that was run at the sequencing end. And so you can see here that this happens to be my reverse primer. These are the Fulmer primers from 1994. This primer did not successfully amplify the gene that I wanted it to. In fact, there's no PCR product. And so you can tell that by looking at both the quality scores as well as the contiguous read length, meaning how many base pairs were successfully sequenced and returned to you as a result. So I know that the amplicon that I'm aiming for is around 600 to 700 base pairs in length. And so most of these were very successful. I'd say I'm pretty happy with these results. The quality scores are good, except for this one, of course, which was not amplified. And so this sometimes happens when you run your bands on the gel. You can't always tell that both primers functioned properly in attaching to the sequence of interest. And so in this case, the reverse did not work, but the forward did. And that's why I always suggest to people that I'm instructing to sequence both the forward and reverse directions, not only to create a consensus sequence ultimately for use in analyses, but also in case one of them does not amplify, then you at least have something to look at. And so the deliverables that are returned to you are three different types of files. You have your FASTA sequence file, your trace file, which will be used to visualize a chromatogram of the read, as well as the phd.1 file. And so all data is better than no data. So I always suggest to select everything. I'm personally going to deselect this one just because I can already tell that that didn't work. And if you go up to the top here, you can select which of those three sequence files you would like to download. In my case, I always download all three. You can also create a multi-FASTA sequence file, which puts all of the sequences obtained into a single file. You can also export the quality scores and contiguous read lengths as well. However, I personally just write these down in my lab notebook because it's just easier for me that way. And once you're ready, you can hit download, and those will be put into your download file as a .zip folder. So what you'll do is you will extract those files. It creates three different folders. It puts all of your FASTA files in one folder, all of your trace files in another, and all of your PhD1 files in another. So they're kept separate. And so you'll have all of the individuals in each one of those folders. Okay, so the next step that you're going to want to do is you're going to want to verify that the sequences that you've gotten from Genewiz are actually from the taxa that you are interested in. PCRs have been tricky to many people in the past, right? There's the infamous instances where people sequence themselves. I have personally sequenced erroneous bacteria, flies, moths, particularly with those universal primers. And so you don't want to spend time cleaning and aligning sequences that aren't of any use to you. So we are going to conduct a basic local alignment, the search tool within NCBI. And so we're going to conduct a nucleotide blast, which will search the NCBI database, NCBI being the National Center for Biotechnology Information. And we're going to see if the sequences that we've got are the ones that we are targeting. So we want to go to the DNA And I'm going to go into the sequence folder. So the AB1 are the trace files, which we'll look at later. The sequence files are just the raw sequence files. So there's no cleaning that's been done. And at this point, you don't really need to clean things in order for BLAST to function. So I'm going to select this first sequence. I'm going to open it. And everything here can pretty much stay the same. You can select which database you want to search against. I am personally interested in the nucleotide collection. And I'm going to search for highly similar sequences. Okay, so then I am going to hit BLAST, and I will be back when I get the results. Okay, so the BLAST results are in. So you can see here that the molecule type is DNA. The query length was identified as 755 base pairs. And this is what the results page looks like. So in particular, these are ordered by percent identity. And so you can see that the highest percent identity is up here at 88.82%. And this is Desperecia. This is a genus of snail, deep sea snail to be exact. And this is actually the genus that I was hoping to find. So I can say with relative certainty that I have at least gotten the sequence within, a reasonable within a reasonable taxon. I haven't erroneously sequenced, a moth or myself or my cat, So with that in mind, we can move on to the next step, which is going to be loading our sequences and cleaning them in the program called BioEdit. BioEdit Sequence Alignment Editor is a free program. You can download it free from the internet. It's a great little program and can do a lot. And so what I've done is I've already opened a file of interest. So this is one of my AB1 files. You can see that extension here. When you open an AB1 file in BioEdit, both of these windows automatically open. You get both the sequence read as well as the visualization, of your trace file. So this is called a chromatogram. And this is essentially the data that the sequencing company obtained and is looking at when they are giving you your sequence read back. And so this is a really good way to visually assess the reliability of your data. So you can scroll through this, You can scroll through this, and each of the peaks represents a base call. And so you can see the resultant read at the top here. So they're color-coded, so it makes it very easy to read. And you can also see if there are areas such as this position here, where there's some unreliability. Perhaps this base call is a little bit, not super confident. And so what I do is I typically zoom in. So if you adjust this vertical scale, it adjusts the height of the base calls. It usually is pretty good at determining the automatic height that it's set to. But if there's ever a read where I'm really kind of trying to figure out what happened, it's useful to know that you can zoom in that way. And so what I do is I always scroll all the way to the end, and I go through my sequences and truly edit them by hand. I am sure that there are faster ways to do this. Personally, though, I like being able to have my hands on things. And this is the way that I was taught to do it. So that is the way that I am teaching you to do it. So the reason why I start at the end of the sequence is because as you go through and verify the base calls, it's best to not mess with the base position. If you start to trim your sequence from the beginning, none of your base pairs will ever match, or your base positions will ever match. it just makes it a lot harder. There's no reason to complicate things any more than they already are. So I always recommend starting from the back and working forward. And so to start, typically what you want to do is most sequences will have this very messy end at the end of the sequence, usually following the primer sequence that you used. And so I know that typically the primer sequences typically have, a repeating base. So in this case, it's this block of repeating T's from about 701 or 5. Yeah, for 5 positions. And you can see that actually down here. I should have done that, sorry. Okay, so what we're going to do is I actually don't want anything after this point. So after 701 bases, I actually just want to trim that right off because this is pretty low quality and it won't help me in my analyses. And so the first thing you want to do is you want to identify this mode here. Right now it says select/slide. I need to change that to edit in order to edit the sequence, okay? Then what you can do is you just click right onto the sequence. You can drag to select the area of interest, and I am just going to hit delete. So that part of the read is now gone from this sequence, okay? And now what I'm going to do is I am going to go through, and usually if they've called a base, it's with good confidence that that is the base of interest. However, there are usually some areas where I'm like, I don't know if I agree with the program's call on this. And so let's see if I can find, yeah, so here. So this looks like it is at 290, around 290. So let's go there and see if we can find what base this is talking about. Yeah, so this is G here, 296. I don't particularly agree. So I'm actually just going to select this base here. And I don't know if I particularly agree with this base call. I am going to, instead of changing this to a C or a T or something else, you can change it to an N. And so if you highlight a certain letter and you can type in a different base, so you can change any of these bases that you'd like. Of course, you don't want to manually do that too often because you don't want to manipulate your data any more than is absolutely necessary for a variety of reasons, particularly academic integrity. And so the reason why I have inserted an N means that this is an unsure base call, means that I cannot be confident what base this actually is. But this is where the reverse read comes in handy, is you can then see what the reverse read says, and perhaps you can actually fill this in using the read in the other direction. So what I've done is on a separate sheet of paper or a Word document, I always keep track of, okay, base 291, I've inserted an N, it was either a T or a G. And then when I go back to the reverse read, I can see which one is most likely the real base call. If I still can't tell, leaving something as an unsure base is perfectly acceptable. Your phylogenetic analyses will still run just fine. And so most of these seem all right to me. Some are more messy than others, of course. You have a lot of background here, but you can still see that the most likely call is the one that they chose, which is G here. And so yeah, this actually looks all right to me. So this is not a super messy sequence, thankfully. Just like in the at the end of the sequence, the beginning of the sequence is also typically pretty messy, which you can see here. And so we also want to trim that off as well. You don't have to keep track of exactly what you're trimming to make the reverse read match. that will all come out when you align your sequences with each other. And so I am going to call it probably at base position 31. Okay, so everything before 31, I'm also going to trim. And so now I have my sequence, okay? So what you want to do now is I'm actually going to copy this whole sequence, okay? So I'm copying it, and I'm going to open another file. So this is from the same day. This is the reverse, okay? And so I have my chromatogram. This is the forward one. I'm going to close that. This is the reverse read. And this is the reverse sequence. So what you want to do is, oops, sorry, is I'm right-clicking on this region on the left here. And I'm going to say paste sequences. And that did not work. So I think what I actually have to do is copy sequence, paste sequence. There we go. Okay. And yeah, so you can see there's no ambiguous sequences, bases at the beginning. So it has successfully copied over my trimmed file here, right? You can verify that visually. And so one thing I would recommend is to never save the changes to this raw file. We will export these sequences separately, but never save those changes. Save changes to file untitled? Absolutely not. You always want your raw sequence file to remain intact. The data that you get from Genewiz when you sequence something is only available for 2 years. And so particularly if you're doing a PhD or a postdoc, you are usually not done with your project in 2 years. So you want to save that raw file intact. So I did not save those changes. So now we have our forward read. Now we're going to be looking at the reverse read. And so eventually, the next step after editing is going to be reverse complementing this read. But for now, we're going to verify the base calls, again, using the chromatogram. So you can already see that this read is much cleaner than the other one. There's a lot less background noise. These base calls are very confident, so I feel good about this sequence. But just like before, you want to start at the end and trim the sequences. So I can already see that we've got some repeats of triple A's here. And so I'm not super thrilled about how all of this from here on out looks. Yeah, so I think, again, this is your call, and this is not make or break. It's always best to keep more than less. And so after these 3 A's at position 661, right here, I am going to delete the remainder of this sequence. So I'm just selecting this sequence here. And again, you need to change the mode, because otherwise, if you leave it at select/slide and you hit delete, nothing will happen. So you hit edit, and I'm going to delete that part of the sequence. Okay. And so I can already tell that this sequence is going to be shorter than the forward, and that is okay. And so I'm looking through, seeing if there's any ambiguity in these base calls, but I think this looks pretty good to me. Okay, so once again, we're going to go to the front of the sequence. We're going to delete the ambiguous regions. So everything looks honestly pretty good. Literally until it doesn't. So I'm not going to delete more than I have to, so I am just going to delete the unsure part. So I am at this point going to select this sequence only. And I am going to reverse complement this sequence. And you can do that using the sequence options up here. You can go to nucleic acid and hit reverse complement, but you can also see here that there's a shortcut embedded within the program. If you, oops, sorry. If you select, so you want to verify that you've selected the entire sequence, which we have. If you hit select Control R, you can see that that has reverse complemented the sequence. So if I hit undo, redo, undo, redo. Okay, so this is reverse complemented now. It's not going to be, you'll notice, it's not going to be an exact match to the forward read right away because these sequences are not aligned with each other. That is going to be the next step that we do. Okay, so I'm going to save this sequence and I will show you, or sorry, these sequences into a new cleaned folder. Again, never save these changes to the AB1 files. And I will resume the tutorial in just a minute here once I get everything set up. Okay. Okay, so we have opened the next program that we're going to use. This is called Mega. So Mega is also a free software that you can download directly online. It is short for Molecular Evolutionary Genetics Analysis. And we are going to use this to actually align our sequences. And so this is the window. This is the main page of Mega. It's a very nice graphical user interface. So there's a lot of tutorials online, and honestly, it's relatively intuitive to learn. But of course, I'll walk you through everything. So what we are going to do is I have saved those sequences, the cleaned sequences from BioEdit into a separate cleaned folder, as I said. And so now we are going to align them. So at the top here, you hit align. We are going to edit slash build an alignment. And so a little window will pop up, which I don't think shows up in the Zoom screen, but it will tell you to select an option to either create a new alignment, open a saved alignment session, or retrieve a sequence from a file. And so I always start with a clean alignment. So I say create a new alignment, and then you hit OK. It will also then ask you, actually, I think I could probably switch which screen I'm sharing. Here we go. Okay, so it will ask you, I'm not sure if this pop-up shows up, data type for alignment, whether you are looking at DNA or a protein sequence. We are looking at DNA, so that is what I'm going to select. Okay. And so this is what opens. It's just truly a blank alignment. There's nothing in here at all. And so what you want to do is at the top, under Edit, you want to insert sequence from file. And so this is the cleaned sequence file from BioEdit. When you go to BioEdit and you hit Save As, after you're done editing everything, it automatically saves as a FASTA file, which is readable by a number of different programs. And so I'm going to select this FASTA file. I'm going to open it here. I'm also going to select this blank default sequence that opens because there always has to be at least one sequence in the file. I'm just going to delete that one. But we have our two edited sequences here. And so it's important to keep in mind that the reverse sequence has already been reverse complemented. So we don't have to do anything with that. So in order to align the sequences, you're going to go to the top here under alignment, and you're going to select what alignment program you want to run. So I personally, for statistical reasons, always align by Clustal W. But I'm sure that your advisor and your personal views, have opinions about these. So do what you think is best for your research. Personally, I'm going to align by cluster W. A little window pops up that says nothing selected for alignment. Select all. I will hit OK. And hopefully this shows up, but if it doesn't, an options window has appeared, which allows you to adjust the gap opening penalties as well as the gap extension penalties. I typically use the defaults embedded within cluster W. So I don't change anything. I just hit OK and it runs the alignment. Now, of course, as you can see, we only have 2 sequences here. So an alignment takes no time at all. But if you're working with, a really big data set, yeah, this alignment can take quite a bit of time. So allow yourself time for your program to run. Okay, so you can see here that as you scroll through, yeah, the sequences match, which is great. That's what you want to see. This is where you can look at differences in alleles. For instance, there's this difference here. There seems to be a deletion in the reverse sequence, but not in the forward. We can find the unsure base call that we identified in the forward read. And if you recall, it was unclear whether the base was actually a G or a T. And so because we saw how clean the reverse sequence was, I feel confident in this base call here, also considering that it was between a G or a T for the forward read. And so I am going to retroactively remove that unsure sequence by replacing it with a G. And so at this point, you can either leave your sequences like this, or you can make what is called a consensus sequence, which is where you combine both of your sequences together. And so that would be, for instance, if I filled this in with a C, if I filled in the gap that was over here with a T, and if I filled in the beginning of the sequence here, so that they all match. And then what you can actually do is you can rename this to be the consensus, and you can actually delete the other one. And that just keeps your alignments, your phylogenetic analyses a lot smaller and a lot more efficiently run. And so that's what I would recommend. Ideally, after you input all of your sequences, you would have quite a big sequence alignment file here. And once I do that for all of my sequences, part 2 of this little tutorial will be looking at how to actually run phylogenetic analyses using both the maximum likelihood statistical method embedded within Mega, as well as the Bayesian statistical method embedded within the program Beast and Beauty. Okay, so until then, hopefully this was helpful, and let me know if you have any ideas about how I can improve my pipeline. I'm all ears. So thank you so much for watching.